The overall goal of the following experiment is to use optical trapping combined with spinning disk confocal microscopy to observe host-pathogen interactions in real time. This is achieved by first adding the pathogens of interest to a chambered cover glass. Next, a particle is selected and captured with the optical trap. Finally, the particle is directed to the cell of interest. As seen here, optical trapping can be an effective method for controlling two particles for observation of dynamic intercellular interactions.
This method can answer important questions in host-pathogen interactions in the fields of infectious disease and immunology, including what is the effect on phagocytosis when complete control of spatial relationships between host cells and pathogens are obtained, as well as whether the order of pathogens being taken up by a phagocytic cell affect phagosomal maturation.
Generally, individuals new to this method will struggle because trapping conditions have to be optimized for different cell and bead types.
Visual demonstration of this method is critical, as the process to set up and integrate a spinning disc confocal to an optical trap is difficult to conceptualize due to the precise alignment that is necessary for all the optical components.
Harvest the pathogen of interest. For example, here 300 microliters of Candida albicans grown overnight in broth is removed from culture and transfer the pathogens into a 1.5 milliliter reaction tube. Then wash the pathogens 3 times at approximately 1300 Gs for 5 minutes at room temperature.